So we talked about the blood plasma. So that's going to be this yellowy kind of watery portion up here at the top. Then at the bottom part, we're going to have the formed elements. So the formed elements are going to be all the cells, as well as the cell fragments, which are made up of the red blood cells, white blood cells, and our platelets. Okay. So this red portion here is going to be made up of our red blood cells only. So that's going to be about 45% of your total blood volume. Okay. In between the blood plasma and the red blood cell component, there's going to be a small little layer right here in the middle, and that is called the buffy coat. And that is where we're going to find all of our white blood cells and our platelets. Okay. So whenever you spin your blood, it actually ends up separating all of the entirety of your blood in your blood vial, and then you'll end up seeing the white blood cells, the blood plasma, as well as the red blood cells here. Okay. All right, so when we look at the different components of the blood, the first component that we're going to look at is the blood plasma. And so the blood plasma is going to be made up of about 91% water. So the majority of it is going to be water, and then about 9% solutes. And those solutes are typically going to be made up of plasma proteins like albumin, globulins, and fibrinogens. You don't need to worry too much about what those are right now. And then in addition to all of those plasma proteins, there's also going to be a bunch of electrolytes like sodium ions, chloride ions, potassium ions, like things like that. Any of those electrolytes, various nutrients, enzymes, hormones, and waste products are all going to also be found inside the blood plasma itself. Okay, so all that's going to be dissolved in there. Okay, and then again, making up the formed elements, we're going to find our red blood cells like we see over here. We're going to see some white blood cells, and then our platelets over here. Okay, so when we look at a smear or a blood smear slide, okay, and you guys have seen this already in lab already, and so you're going to see a bunch of red blood cells all scattered throughout your slide here. You're going to see a number of white blood cells like we see all over here. And then you're going to see some of these smaller little components, these small little dots. These are all of the platelets. So platelets, as we'll talk about at the end of this lecture, there's going to be these large cells that eventually will split and break up. And so those breaks, or those kind of bursting of those cells, are going to form these platelets here. And then all of this white space surrounding all of our cells here isn't just empty space. That's the actual blood plasma within your blood. Okay? So those are what everything kind of looks like there. We've kind of looked at it already. And for lab, you guys are going to be taking a look at those in a little bit more detail as well. Okay. So when we're looking at blood, there's one specific measurement that's going to be pretty important. And so that measurement is going to be hematocrit. So the hematocrit tells you the percentage of your total blood volume that is made up by your red blood cells. Okay. So in females, the hematocrit level should be about 42%. So if you take their entire blood volume, 42% of that should be made up of just red blood cells. In males, the hematocrit level should be about 47%. So again, there should be about 47% red blood cells in the total blood volume. Now, those numbers are really, really important because those will tell you, or those will give you an indication of what type of condition someone might be dealing with. It can tell you if they're dealing with anemia. It can tell you if they're dealing with different types of polycythemia, things like that. It can give you an indication of different types of conditions that person is going through. Okay. The first kind of condition here that we're going to be taking a look at is hypoxia. Okay, so hypoxia is a situation where you have oxygen deficiency within your red blood cells. Okay? And so when someone is dealing with hypoxia, that is going to trigger your kidneys to start producing new red blood cells. Okay? That's going to trigger, so in order to do that, in order to produce new red blood cells, the kidney is going to start undergoing, or start releasing a hormone called erythropoietin, or EPO. Yeah, and that EPO will help to stimulate red blood cell production. We'll talk a little bit more about that as we go forward here. Okay. And so that'll happen when you're exposed to hypoxic conditions. Okay. And so when someone is undergoing various conditions like hemorrhaging, red bone marrow failure, for example, or red blood cell destruction, that can cause the hematocrit level to drop. Okay. And so it can cause the hematocrit level to drop because if you're dealing with a hemorrhage, you have individuals bleeding out, so you're literally losing red blood cells. Okay? And so as a result, your hematocrit level is going to shrink and drop a little bit. Okay? If you're dealing with red blood cell destruction, again, now you have a smaller component of your red blood cells that are making up your total blood volume, so your blood volume is, or your hematocrit level is going to drop in that capacity as well. Okay? So those are different types of things that can lead to anemia. Okay? And so anemia is where the blood has a reduced oxygen carrying capacity. It's not able to carry as much oxygen as it should be able to. Okay? The opposite of anemia is what's called polycythemia. Okay? So in polycythemia, instead of having a drop in hematocrit level, you have an increase.
It started off, it just automatically had my video on, and I was like...
Oh yeah, my bad. I'll edit that out. You'll be fine. Yeah, so the polycythemia is where you have an increase in your hematocrit level above 54%. Okay, so and so this can be due to a number of different reasons. This can be due to whether you have an increase in red blood cell production or an unregulated increase in red blood cell production.
I want to know how long I have.
So if you're constantly producing new red blood cells, that's going to cause your hematocrit level to increase because now you have more red blood cells available. If you have tissue hypoxia will trigger your kidneys to start producing erythropoietin, which will stimulate red blood cell production and again increase your hematocrit level. If you have dehydration, and dehydration will literally be you have a lower blood volume, and so if your blood plasma level drops, your blood cells are going to make up a larger component or a larger percentage of your total blood volume. So that's going to increase your blood volume there, or blood doping too, and we'll talk about that as well. Okay. So polycythemia is a condition that's not really not good because what it does, and I'll talk about this in more detail later, is it's going to start to cause your blood to become a lot thicker. And so as a result, it becomes a lot harder to pump the blood from point A to point B. Your heart has to work a little bit harder, and then it causes your blood pressure to increase and causes a whole lot of other issues. And again, we'll talk about that in more detail too. Okay. Okay. So that is the components of our blood there. Okay. So this is just a little schematic here that shows you kind of how much all of your blood is going, or what is made up of your blood. So if you look at all your body weight, your blood is going to account for about 8% of that. And then 55% of your blood is going to be your blood plasma, 45% is going to be the formed elements, and then it's got further breakdowns from there. Okay, so question here. So which of the following substances listed below are transported in blood? Okay, so we got carbon dioxide, we got various nutrients such as vitamins, glucose, hormones, oxygen, and heat. Okay. Which of those 5 is able to be transported? You can either type it in the chat or you can just say it.
I look ahead, it's A.
I'm hearing A. Do we agree with A?
It's
yes.
A.
Yes, perfect. A is correct. Yeah, so all 5 of those are going to be able to be transported in the blood. Perfect. Cool. So next one. So which of the following is false regarding hematocrit? Okay, so we got A, it equals the percentage of total blood volume occupied by your red blood cells. B, the normal average for females is less than the normal average for males. C, hemorrhage would cause a drop in the hematocrit value. D, an increase in the hematocrit value above 54% represents anemia. Or E, all of these are true. Which one is false?
D.
D, yeah,
D.
perfect. So what would a hematocrit above 54% represent? Polycythemia, right. So above 54%, you're looking at polycythemia. Below that 45%-ish average, you're looking at anemia.
Anemia.
Okay, any questions so far?
Nope.
Yes, I got a question.
Yeah.
So you were saying in a hypoxic environment that would increase red blood cell production?
Right.
Okay.
Yeah. So when you're in a hypoxic environment, you're not able to get as much oxygen to where it needs to go.
Right.
So in order to combat that, what your body will do is it's going to start releasing erythropoietin to help you produce more red blood cells. So his question, for those of you guys who may not have been able to hear, he was asking was about hypoxia and how it's able to produce new red blood cells. So the reason for that is by the kidneys being able to produce erythropoietin to help you stimulate more red blood cell production. Cool. Did you have a follow-up question to that? Oh yeah, go ahead.
Yeah, I was gonna say, is that kind of why, like athletes will go to like Denver or somewhere up high to train under like low oxygen levels?
Yes. Yeah. So his question was, is this why athletes will go to high altitudes to help them to help them essentially, perform better in their particular sport. Yes, and I'll explain why that's the case in a little bit. So stay tuned for that one. Very good connection there. Really good. Any other questions here? Okay. All right, cool. Okay, so we're going to look at how we're going to be able to produce red blood cells. Okay, so the process of producing new red blood cells is this process called hematopoiesis. And so hematopoiesis is where we start to develop new formed elements. Okay, and so this is going to take place inside the red bone marrow. And if we remember from chapter 6, that red bone marrow is going to be found inside the trabeculae of the spongy bone. Okay, now what's kind of interesting is that when you're looking at an infant versus an adult, an infant is going to have red bone marrow all over their body. In every single bone, they're going to have red bone marrow available. So they'll be able to produce new red blood cells all over their skeleton. Now, as that infant eventually gets older, the red bone marrow in the appendages, usually in the arms and the legs, ends up getting replaced by yellow bone marrow, okay, where you start to produce fat. And so essentially, the only places where you end up producing red bone marrow is essentially just in the axial skeleton. Okay, so in the skull, in the ribs, the sternum, and then in the hips too, okay, as well as in the proximal epiphyses of the femur as well as the humerus. Okay. So it essentially gets confined to a specific place because the yellow bone marrow begins to displace and replace that red bone marrow in the infant skeleton in the appendages. Okay. So this process is going to start with specific stem cells inside the red bone marrow called the pluripotent stem cells. Okay. And so pluripotent stem cells are really cool because they have the ability to develop into multiple different types of cells. Okay. So as they undergo cell division, they're going to start reproducing, and sometimes they'll develop into new blood cells, sometimes they'll develop into osteoblasts, and sometimes they'll develop into new muscle cells. But specifically for our situation here, we're going to be looking at how these pluripotent stem cells will eventually produce new red blood cells. Okay. So the thing about these blood cells is that when you produce these blood cells, they're no longer able to undergo cell division, okay, except for lymphocytes. Those are the only ones that are able to undergo cell division. Okay. But red blood cells and most white blood cells, the platelets, they're not able to undergo cell division once they've been produced. So the only way that you can produce new red blood cells is by starting with brand new pluripotent stem cells at the beginning of this whole process here. Okay. So we're going to look at how this ends up playing out here. Okay, so those pluripotent stem cells inside the bone marrow are eventually going to develop into one of two types of stem cells. The first stem cell that they can develop into are the myeloid stem cells. Okay. And so these myeloid stem cells will end up developing eventually into red blood cells, platelets, and then all the white blood cells except for lymphocytes. Okay. So here we have our hematopoietic stem cell. Okay. It eventually will become a pluripotent stem cell, and then from there it can develop into a myeloid stem cell. We'll see develop into red blood cells, platelets, and all kinds of different white blood cells here. Okay. Now, as they develop, all their development is going to take place inside the red bone marrow. Okay. So that's option A for the pluripotent stem cells. Option B is that they can go down the lymphoid stem cell pathway. Okay? And if they go down the lymphoid stem cell pathway, then they will eventually develop into either a B lymphocyte or a T lymphocyte or an NK cell, which is called a natural killer cell. Okay? So these B lymphocytes and T lymphocytes you've probably heard of as B cells and T cells. Okay? Those are some of the really important immune system cells that will eventually help produce some significant immune functions down the road. Okay, so we don't have too much. We're not going to be able to talk about those in a lot of detail because we have to skip that chapter, unfortunately. But those are going to be the two cells that end up getting formed here. Okay. And then the natural killer cells have an ability to break down and destroy lots of different types of pathogens as well. Okay. So when these guys start to develop, okay, they're going to begin developing inside the red bone marrow, and then they're going to finish their maturation process inside the lymphatic cells and lymphatic tissues as well. Okay, so once they decide which pathway they're going to go into, once they become a myeloid progenitor cell or a lymphoid progenitor cell or stem cell, they're eventually going to form some precursor cells. Okay. Those precursor cells are also called blasts, these are where the cells eventually become kind of committed to that particular pathway. Okay. So for example, we might have a myeloid stem cell that eventually becomes a red blood cell precursor, and at that point it becomes committed to becoming a red blood cell. Okay, that's the point of no return. There's no going back at that point. Okay. So once it becomes a precursor cell, it's committed to that pathway, and then eventually will become that particular type of formed element. Okay, does this make sense so far?
Yes.
Yes.
Okay, all right, cool.
Yeah.
Okay, all right, so I'm going to take a quick little detour here. So remember, there's going to be one hormone that we talked about, which is erythropoietin, and so that is going to be released from the kidneys in order to help produce red blood cells. Okay. So if we need new red blood cells to be produced, our kidneys are going to release erythropoietin, and they're going to trigger our hematopoietic stem cells to become the pluripotent stem cells, and then help undergo this entire pathway to help us form our red blood cells, okay, ultimately here. Now, if we need to form new platelets, we're going to release a hormone from the liver called thrombopoietin, or TPO. And so what will happen is we'll produce a cell type called the megakaryocytes, which are not shown here, or yeah, right here. So these megakaryocytes are these guys here. And when these megakaryocytes mature, they will eventually rupture and form these little platelets. Okay. And so that's what we'll do if we need to produce new platelets. Okay. So 2 hormones that are going to be important to help us produce different types of cells. Okay. Well, this is just a little schematic that kind of helps you show, it's more detail than you need to worry about, but this is a schematic that kind of helps show kind of the pathway of all of these different types of cells. Okay, so again, starting with the pluripotent stem cell, it can then either enter the myeloid stem cell pathway or the lymphoid stem cell pathway. If it goes the lymphoid stem cell pathway, it's going to develop into a B lymphocyte, a T lymphocyte, or a natural killer cell. If it goes the myeloid pathway, it's going to develop into any of those other types of formed elements. Okay, so if someone is dealing with some kind of blood condition, what a doctor can do is they can undergo or cause someone to undergo what's called a bone marrow biopsy. And so what that is they'll take this very thick biopsy needle, they'll insert it into the bone, okay, and then try to get into the spongy bone and then extract some of the bone marrow out. Okay. Usually the place where they're going to be doing this is going to be either through the hips, through the ilium, or through the ilium, or through the vertebrae. In either event, it hurts a lot because you actually have to penetrate the bone in order to actually get into the spongy bone trabeculae and then pull out some of the bone marrow. So not a very comfortable condition. It's actually extremely painful. But by pulling out a lot of that bone marrow, you're going to be able to see, the different types of precursor cells and be able to identify, are you producing more of one type of cell than others, or vice versa, and try to figure out what exactly is going on there. Okay. So not comfortable at all, but it's an extremely important process. Okay, so now we're going to be taking a look at the different types of formed elements, and we're going to start with the red blood cells, and this is going to be kind of making up the majority of what we're going to be talking about today. Okay, so red blood cells are also called erythrocytes, and so what we're going to be doing is we're going to look at the anatomy first of the red blood cell, and we're going to look at how its anatomy is going to be able to help it do what it needs to do. Okay, and so its anatomy is going to actually be really cool. So I'm going to nerd out for a little bit, just kind of bear with me, but this is actually like really, really, really important here. Okay, so first thing that we're going to be looking at here is when we look at our red blood cell, it's going to be taking on a biconcave shape. Okay, so it's basically going to look like a donut with a little bit of membrane kind of in the middle, okay, where you would have that hole. Okay, and so that plasma membrane is going to be really, really strong and really flexible, and that's going to allow the red blood cell to be able to maneuver through the blood vessels. Okay, so that's number one. Number two is on the surface of the red blood cells, there's going to be a number of different types of glycoproteins. Okay, those glycoproteins are going to determine your blood type. Okay, and we'll look at that in a little more detail as we go forward here. Okay, so there's going to be some glycoproteins that are going to determine whether you have type A blood versus type B blood, or type AB or type O blood, and then determine, and then the Rh blood group proteins are going to determine whether you're positive or negative. Okay, so the combination of these two proteins are going to help to determine whether you have like A positive blood or A negative blood, or AB positive or O negative, and things like that. Okay, so we're going to be taking a look at that in a little more detail as we go forward here too. Okay, The third thing that's going to be really, really important here is that these red blood cells are not going to have a nucleus, and they're not going to have any organelles, or very few organelles, if anything. Okay. That is going to be really important because these red blood cells, because they don't have a nucleus, are not going to be able to undergo cell division. Okay. So you cannot produce red blood cells from other red blood cells. Okay. Again, you have to go through the whole process of starting from these, from the pluripotent stem cells, and then again go through that whole myeloid pathway to eventually produce new red blood cells. So you cannot produce new red blood cells from other red blood cells. Okay. The other thing is that because there's no organelles, those organelles are not going to, because you're not going to have those organelles, you are not going to be able to undergo various types of metabolic activities through the red blood cells. Okay. And then last thing is that they're going to have lots of hemoglobin. Okay, so hemoglobin is going to be a protein that's going to be important with actually carrying oxygen to where it needs to go. Okay. So all that being said, all of those different components, with the exception of the glycoproteins, those are all going to be important for allowing blood to be able to transport oxygen with a really high ability to do so. Okay. So starting with number one, so the absence of a nucleus and any organelles. Okay. So because there's no nucleus and there's no organelles, there's going to be all kinds of space for activities. Okay, you kind of think about that the movie Step Brothers, where all kinds of room for activities. Same thing is going to be happening inside of our red blood cells. Because there's no nucleus and no other organelles like other cells do, it's just going to be pretty much just an empty space. If we think back to chapter 2 when we were looking at cells, they're going to have a nucleus, they're going to have mitochondria, they're going to have the rough ER, the smooth ER, the Golgi, the lysosomes, all kinds of different things making up the cell to make it really busy. Okay. Because the red blood cell doesn't have any of that stuff going on, it's just going to have all kinds of extra space available to hold as much oxygen as possible. Okay. So that's number one. Okay. Number two is because there's no mitochondria, it's not going to be using up any oxygen to produce ATP. Okay. So if you kind of think back to Bio 10 when we're looking at aerobic respiration uses up oxygen, okay, in order to help you produce large amounts of ATP. Okay. Now that's important because mitochondria, if red blood cells were to have mitochondria, okay, they would be using up the oxygen that they have to produce ATP instead of transporting it to where it needs to go. Okay. So by not having mitochondria, it is now in a position where it's not going to be using up any of the oxygen to help itself survive. Okay. It's just going to be using anaerobic respiration. It's not going to be using up oxygen to help produce ATP. Okay. And then the last process here or the last kind of function here is that the biconcave shape, okay, because it's shaped like a donut, that actually increases the surface area of the red blood cell. And so as a result, it's going to be able to pass oxygen and nutrients as well as carbon dioxide and waste a lot faster than it would if it was more like a spherical shape or more of a rectangular shape. Okay. So by having that shape, it's able to pass things across its membrane a lot quicker than other cells would be able to. Okay. You guys follow me so far? Does this all kind of make sense?
Yes.
Yeah.
Yes.
It does.
Yes.
All right.
Like a
Any
review from
questions here?
bio.
Any questions?
No.
All right, cool. Okay, so those are red blood cells, really, really important. So they're going to be able to transport oxygen. Okay. So when we look at each of these individual red blood cells, okay, each of these red blood cells has within it 280 million hemoglobin molecules, hemoglobin proteins, which is pretty incredible because if you remember looking at those red blood cells at the beginning of the semester, okay, those were incredibly small. And just think that inside each of these individual tiny red blood cells, you're going to have 280 million proteins that are just geared for carrying oxygen. Okay, and so as they carry oxygen, they're going to actually contribute to the blood's red color. Okay. So when we look at hemoglobin, okay, hemoglobin is going to have 4 different globins that make its protein up. Okay. Here we have one globin here, this light blue structure here. We have another globin, this dark blue. We have a third globin, which is this light blue, and then a fourth globin, which is this this darker blue. Okay. So each of these are globin chains. To make up one hemoglobin, there's 4 globins in each hemoglobin. Okay. In the middle of each of those globins, you're going to have a heme group, okay, which is this guy over here. Okay, this kind of guy looks like a little bit of a saucer. Okay. In the middle of each heme group, you're going to have this iron ion. And that iron, it looks like this thing is going to make me kick me out again in 10 minutes. So hopefully it doesn't. Okay, sorry. So this heme group is going to, this iron group is going to be what actually allows you to carry oxygen. Okay, so the iron itself is what carries oxygen. So because you have 4 irons per hemoglobin molecule, you're able to carry up to 4 oxygen molecules at a time. Okay, okay. So hemoglobin has the ability to release oxygen as well as transport carbon dioxide as well. So when that once the red blood cell gets to a particular type of tissue, the hemoglobin is going to unload and release that oxygen into the interstitial fluid, and then from there, that oxygen is then going to diffuse into that particular cell. Okay. When that cell uses up that oxygen, it's going to end up forming carbon dioxide, and that carbon dioxide will again get picked up by hemoglobin, get transported to the lungs, and then we're going to be able to exhale that carbon dioxide. Okay. Okay. So when we look at anemia, so anemia is going to be a lack of oxygen carrying capacity. So you're not going to be able to carry as much oxygen. Okay. And usually this is going to be due to a number of different reasons. It can be due to having enough or not enough iron in your red blood cells or within your hemoglobin, or it could be due to a number of different factors in at any other type of the pathway, aspect of the pathway. Okay, so people who are anemic will usually have they'll usually be pretty fatigued and pretty tired very quickly, and that's because they're not bringing in enough oxygen. They're not able to produce as much ATP as a result, and so because they're not able to produce as much ATP, they end up getting tired very quickly. And then they're also able to not have as much heat, okay? And so they end up being more cold again because they're not able to produce as much ATP. Okay. So there's various types of anemia. We have iron deficiency anemia, where you just don't have as much iron in your body. There's hemorrhagic anemia, where you're losing red blood cells. And then there's also like sickle cell anemia. Okay? And so with sickle cell anemia, what happens is the hemoglobin inside the red blood cells are not built properly. Okay? And so because they're not built properly, it causes the entire red blood cell as a whole to take on a sickle shape, okay? Which is a problem because that reduces the surface area. It's not able to deliver as much oxygen. But more importantly, the sickle-shaped cells also begin to link onto each other. Okay? And so if you have a bunch of sickle-shaped cells all linking onto each other, they're going to end up forming these large clumps of red blood cells, okay, that cannot like just release each other very, very easily. And so as a result, they end up forming these clots and these clumps within your blood vessels, and then they can cause obstruction of your various types of blood vessels and prevent blood from getting to where it needs to go. Okay, so sickle cell anemia is one of the more detrimental types of anemia that people can be dealing with. Okay, all right. So now we're looking at red blood cells. Okay, red blood cells are only going to live for about 120 days, so about 4 months is their lifespan. Okay, the reason that they live for such a short period of time is because of significant wear and tear of their plasma membranes. And so as they run through their capillaries, the capillaries are only about as big as the red blood cells themselves. Okay? So in order for red blood cells to pass through, they have to pass through in basically a single file line. Okay, so if we kind of look at this bottom picture here, we can see these red blood cells are having to fold in order to fit within these capillaries. Okay? Which again is not really a problem, but eventually kind of becomes a serious issue because the red blood cells start to cause lots of abrasions or develop lots of abrasions as they run through the blood vessels. Okay, so as they develop all those abrasions, they start to destroy kind of the plasma membrane, but they have no way of repairing themselves because they don't have any nuclei or any organelles. Okay, so the damage that they get is just what they end up with, and once they die, there's no way to repair them. Okay? So for that reason, they only live for a very short period of time, only about 4 months or so. So eventually, again, if you need to replace those red blood cells, they're going to be replaced through that process of erythropoiesis, which is where you produce new red blood cells through the red bone marrow. The really cool thing about erythropoiesis is that it happens at about the same pace as red blood cell destruction. Okay? So you're producing new red blood cells at about the same rate as you're losing red blood cells. Okay? So in a normal individual, you're never going to have a situation where you're producing more red blood cells than are dying, or having more red blood cells dying than you're able to produce. It's always going to be in the balance, happening at about the same pace. And so erythropoiesis is going to happen, again, for a number of different reasons. Okay? It can be due to hypoxia. So again, if you're in a low oxygen environment, your body's going to start producing extra red blood cells through erythropoiesis in order to help take in as much oxygen and use as much oxygen as is available to you, and make the most of it, what's available to you. You can have erythropoiesis take place if you're anemic. Okay, so if you don't have as much red blood cells, or if your iron levels are low, you're going to produce new red blood cells to help make sure that you're using that oxygen as effectively as possible. If you have any circulatory problems, if you have, like, for example, an obstruction in one of your blood vessels, you might have extra erythropoiesis take place to help you make sure that you're able to transport oxygen to those particular areas. And again, if you have any tissue hypoxia, erythropoietin is going to get produced in order to help kickstart this whole process here. Okay, so this kind of goes back to that question earlier about blood doping, or in some cases where athletes may train at higher altitudes to increase their red blood cell production. Okay, but we're going to talk about blood doping first, and this could kind of work exactly the same way as training at higher altitudes. Okay? And so essentially with what blood doping is whenever you have some kind of athlete who wants to increase their muscles' ability to perform and kind of reduce the ability of their muscles to fatigue, they'll start doping themselves with various types of substances, usually erythropoietin. Okay? And so that's going to jumpstart their process of producing extra red blood cells. Okay? By producing more red blood cells, you're able to deliver more oxygen to those particular muscles during those athletic events. And by delivering more oxygen, that's going to reduce the chances of those muscles of fatiguing. Okay? And so you're going to be able to delay the onset of fatigue and be able to improve the ability of that athlete to perform at that particular event. Okay? And so this is what Lance Armstrong was found to have been doing for a number of different periods of time while he was doing lots of these bicycle marathons. Okay? So he would kind of blood dope, increase the amount of red blood cells available, and as a result, his muscles were not fatiguing as fast, and he was able to perform a lot better. Okay? And so that is where we end up with polycythemia, where you have an increase in the red blood cell production. Okay? Now, while this does help increase your ability to perform, this is also really dangerous because this causes the blood to become a lot more viscous. And instead of moving like water, it eventually moves more like syrup. Okay? Now, because the blood becomes a lot thicker, more resistant to moving, the heart has to pump a lot harder in order to pump that blood to where it needs to go. Okay? Because it has to pump a lot harder, that increases the blood pressure, and then it also increases the risk of stroke. Okay? So this is actually not really something you really want to be doing because there's a lot of potential side effects that could go wrong with this. And any individual who does go through this whole situation of blood doping usually has to go through it under the supervision, very close supervision of a physician to make sure that none of these processes end up happening. Now, training at higher altitudes basically goes through this whole same process because, again, you're training in an area where you have less oxygen. Your body's going to be naturally producing red blood cells. And again, that'll help you to be able to perform. Okay. So same process, but the ethics and processes of it are a little bit different here. Any questions here? No? Okay. All right. So now we're going to be taking a look at these blood types. Okay. So when we're looking at these blood types, so you guys some of you guys probably know your blood type. For example, I'm B positive. Some people might be A negative, O negative, AB positive, things like that. Okay. Your blood type is going to be determined by the specific antigens on your red blood cells. And these antigens are just different proteins on your red blood cells. Okay. So for example, if you have type A blood, you're going to have a lot of the type A antigen on your red blood cells. If you have type B blood, you're going to have the B antigen on your red blood cells. Okay. If you have type AB blood, you're going to have both the A and the B antigen on your red blood cells. And if you have type O blood, you're going to have no antigens on your red blood cells. Okay. Now, depending on the type of type of antigen that you have, your body is also going to be producing antibodies against whatever you don't have. Okay. So this is going to be a situation where you're going to be able to kind of protect your body against foreign